Okay, so hello everyone. Thanks to the organization to make this talk possible. So I'm Sébastien Gradit, second year PhD student at the Pasteur Institute, and especially in a Roman Kozul lab called Spatial Regulation of Genomes. And today I will talk about BERG, so a statistical inference methodology to assess a repeated sequence position in ice maps. So first of all, I would like to introduce the subject by showing some drawings made by Fleming in 1882 about Lilium core theorem. And as you can see, the genome folding is not completely random. And as you probably know, we have to fold a 2-meter DNA polymer to fit in a 10 micrometer nucleus of human cell. And that's kind of the same for the yeast. So an 8 millimeter DNA polymer to fit in a 2 micrometer cell nucleus. And the main question that we're interested in is about the genome folding. So it's kind of a chicken and egg problem about does the folding imply the biological processes such as DNA repair, chromosome segregation, and transcription, or is it the contrary? And to answer those questions, we use IC technique. So I'm not going to enter too much into details, but the idea is to form a chimeric reads by fusing loci that are close together in 3D, basically. And we use to represent the data as to contact matrix. So one pixel is representing part of the genome, like bean, for example. In this case, it's the chromosome 16 of the baker yeast, Saccharomyces cerevisiae. And the more the value is high, the more the two loci are able to do contacts at the scale of a cell population. Sorry. And because it is relying on chimeric reads, like some are going to map at multiple positions, and in such case, we don't know how to place the pair on the map. So classical pipelines are discarding such reads, and it appears as white stripes on the maps. So we have mainly 3 chimeric reads configuration. First one, at least one read of the pair is not mapping, so the pair is discarded. The second scenario is that the 2 reads of the pair are mapping only once, so that's okay, we can build the map on. And the third case is that 1 read of the pair is mapping at multiple positions. So we don't know, like in the top example, which is the right coordinate of the blue part of the chimeric read. And such events represent about 40% in Vibrio cholerae, for instance, about 15% in the Saccharomyces, Saccharomyces cerevisiae yeast, and about 35 to 40% of human IC reads. And these are the reads that we are interested in. So the idea is to get a statistical model to assess the right position of the chimeric read. So the idea is to know which were the loci that were in contact in 3D. And to answer that question, we use some statistics extract from the IC protocol. So we have several configurations of the reads, like dangling ends, self-circularized, and mirrors. They are mainly based on the orientation of the reads after the alignment. We are also using the coverage over the genome, like the more the genomic spot is covered, the more likely the reads are coming from this position. And we also use what we call D1, D2 metric, based on the distance to the closest restriction site to answer the question about which of all the plausible pair is the right one. So the idea is to build a map which is representative of the statistical model. And in this example, we have the two blues, which are the plausible position for the first mate, and the red, which is the only one for the second mate. And the idea is to place the pair on the map and to get probability distribution and to draw the right pair from this distribution. So iceberg will be released soon as a Python 3.11 package. It accepts standard file format from IC experiments, and computational times are genome length and richness of repeated sequence variable, but still it took 40 minutes to process data from E. coli and 2 hours for Saccharomyces cerevisiae, respectively 26 million and 47 millions of reads. So so far, we need to evaluate the pipeline, and because we are inferring something that no one even know, what is the ground truth, we had to set a strategy. So the idea is to get the part which is not ambiguous and to split it in two, an outer set to learn the statistics and an inner set to infer, after introducing some ambiguity by duplicating the reads from the alignment files. Then we reconstruct this, let's say, missing part, and we assess the quality of the reconstruction. And here are the first results. So we use a Pearson correlation to get an idea of the quality of the reconstruction. And as you can see, the random model is not that good, which is expected. And if we rely on the coverage of the genome only, it's not enough. But as soon as we are using PFS, which is the polymeric law, we reach a very high score. So we could be very confident about our reconstruction. And when we are using the full model, it's kind of the same. So we can say that the polymeric law is basically carrying all the information. And we tried some other experiment by like creating fakes, duplicated elements, and we let vary the distance between the two, let's say, synthetic repeated sequences, or the number of repeated sequences on the genome, and performance are quite satisfying so far. So now we'll show 3 examples of reconstruction. So the first one is on E. coli DHB10, which have been engineered to store plasmid initially. And it appears that this strain acquire tandem duplication about 100 kb. And on the left, you have the native map. And on the right, you have the reconstructed map. And it looks like what we expected, like the two parts are behaving the same. So it was expected. More interesting is Saccharomyces cerevisiae, so the yeast, and especially on chromosome 12, because it has been observed through microscopy that during the aging of the yeast, some parts of the ribosomal DNA are released in the nucleus and they tend to circularize. And it has been observed also that it tends, such circularized piece of DNA, they tend to contact the chromosome 12 centromere. And it is also observable on the reconstruction on the right. So this is what the blue arrow is meant to point. And to finish, an example on Plasmodium falciparum, which is the parasite of malaria. And it has been previously observed through microscopy and through statistical model projection that the parasite tends to cluster its telomeres with the virulence gene to represent their expression during the parasitic cycle to escape the immune system. And this is what we retrieved after the reconstruction. So here again on the right. So if you take a message, ISBERG should bring a new hypothesis about singular repeated sequences involvement in genomic tridic formation, such as Vibrio cholerae superintegrum, Saccharomyces cerevisiae TY, so retrotransposon of yeast, and Plasmodium falciparum virulence gene repressions. ISBERG may reveal some genome plasticity linked to repeated regions, and it can improve IC normalization and automatic pattern detection on IC maps. And it has to be improved so far for large genome and rich repeated sequences genome. Thanks for your attention. And thanks to the lab, my PI, Romain Cozul, Axel Cornac, my supervisor, and also the French Society of Bioinformatics for giving me the opportunity to present my work. And thanks to you. Oh yeah, if you have more questions, you can join at poster 160, I guess. Yes, 160 to discuss about ISBERG, if you want. And also, if you have questions about the use of French bioinformatics, feel free to chat with me.